was a big part in helping convince the world that it should be done. So I started this organization, a charitable organization, in the late 1990s to be able to, at the time, determine the crystal structures of human proteins and put them in the public domain without restriction. Not many here were alive then, but at the time what happened was, with the Human Genome Project being sequenced, there was a tension between the public and the private sector as to who would own our genome. And the private companies started to want to clone the genes and patent them. And when things are patented, they're hard to use by people. And so the public sector, led by America and the Wellcome Trust, mounted a robust effort to put all the genome sequence information about humans in the public domain for everyone to use. About the same time, the structural biology community started to patent structures. And in response to that, Wellcome Trust and some companies and the Canadian government said, that's silly. Why don't we have an organization that determines the structures of human proteins and put them in the public domain so everyone can use the structural information, the clones to make the structures and everything. So I started that about 2002. And since then, we did a lot of structures, and about 2009 started to change the idea. We've got all these protein structures. We knew the end of structural biology was coming. We didn't know how long it would take, but we knew it was a done deal. Why don't we start to use these structures and try to create research tools for people, and I'll explain the rationale. So fast forward, and our target 2035 is our idea that let's create a potent and selective inhibitor or modulator of every human protein in 10 years. And I'm going to describe the background and the whys now. And so even though we're funded by the pharmaceutical industry, we put everything in the public domain. And it's kind of cognitive dissonance, right? In the next 5 years, we have $70 million from industry to share. And so, and the rationale is what's inhibiting drug discovery is not, can I make a molecule, but can I understand biology so I know which molecule to make? And so the problem in biology is actually us. And so is this a pointer? Yeah. So to show this, I said, okay, why don't we say the human genome is a pretty signature event in human history. We have the code for life, right? And we spent a lot of time and money getting that. With much fanfare, it was announced. And so, but we've kind of under-delivered. So on this axis here is publications, and on this axis here is the gene name. And so you can go into PubMed and say, how many papers are there on kinase 1, 2? So these are the papers before the human genome was sequenced. So we didn't even know these existed, so no kidding. And then we ranked them from the top, most popular to the lowest. You get a power law curve, don't focus on that. The message is a few proteins, papers, a lot of proteins, no papers, okay? So the purpose of sequencing the genome was to uncover the unknown so that we could explain Parkinson's and Alzheimer's and all the things we didn't understand, right? And so here we had it. The template to do this was here. But now you do the same search string, gene and 2017 in PubMed. So you look for Sarc kinase and 2017, how many papers? And you do that for all the kinases, all the genes in the human genome, and you rank them in the same order. And it's kind of stunning and it's kind of sad, despite the fact that these genes have few papers, despite the fact that they've been encoded in us for 2 million years, the human behavior of professors, we don't study them. It's perfectly explicable, though, if you look at the sociology of the academic system. We're not rewarded for discovering stuff, we're rewarded for papers. And try and write a grant or a paper on kinase 512. It's been in the human genome for a long time, I want to know what it does. Where's the garbage? Because that's where your grant is going, right? If you work on this one, even though lysine 32 is so important, it's methylated, and I make the prediction that if it moves like that, the mouse will do this. Oh, brilliant, fund that, right? And so you get this sort of interesting phenomenon. Then you think, well, what about the grant system? The grant system everywhere in the world, I guarantee you write your grants like I write my grants. Background and significance, preliminary data, what are you going to do, risk mitigation. Every grant's the same. We all read the same papers. Every grant's practically the same. right? And so you get this interesting phenomenon where you say, oh, certainly in Canada, oh, we need some American reviewers because it's too incestuous inside of Canada. We need to expand our reviewer pool, which happens. And then you just homogenize review all the way across the world, and you get the same phenomena. This is BRAF. Remind me if I forget to tell you what that is. So now you do the same search string, SARC, and 2017, and Canada, right? It's a well-known fact that Canadians are smarter than everyone. And so what will happen is obviously we're going to be more daring and more not, okay? So you have to normalize it. We publish fewer papers, so that's why it looks noisy. But the pattern is exactly the same. And Bangalore and Harvard and anything, right? We have a very homogeneous system that conspires against doing something. And to be fair, it's really hard to work on a protein. There's no clone, there's no antibody, there's no... If you start to work on that, by the time you've generated all that, your grant's over. And they'd say, where are your papers? So everything kind of makes sense and everything kind of doesn't make sense. So when I went to the pharmaceutical industry, they say, well, if we triple the NIH budget, or if India all of a sudden, in the fantasy world, says we're going to spend $10 billion on R&D, you're going to get this to the extreme. You're not going to see much of this. Like there's 10,000 papers in the human genome with fewer papers than fingers on my hand. Yeah? Okay, so that's why I went to pharmaceutical industry. I said, the problem in drug discovery is we don't understand human biology. We don't understand human biology in part because we don't study half of the genes. And as a reductionist, you know they have to be doing something. So that was the problem statement. And so how do you get to the answer? And so the answer actually comes from the pharmaceutical industry again. So Tim Wilson at GSK was part of this field, the nuclear receptor field, and his group did something interesting. But I'd like to show you the data first. So this is a protein family, a transcription factor family, where the DNA binding domain is part of a two-domain protein where you have a hormone binding domain. So when the hormone binds, the transcription factor changes its shape, recruits the mediator, and off you get transcription. And you've heard of these. That's the estrogen receptor, the androgen receptor, progesterone receptor. And so you see in the early, before 95, when they were all cloned, there was a lot of activity on these ones. And then starting from 95, when all the genes became available, you go to 2009, you say how many papers were on them, and you can see something curious happened. There was a shift in research activity, much more significant than the kinases, to new ones. So we thought, oh, wow, what caused that? Were they cloned first? Linked to human genetic disease? No. This one's mutated in mature onset diabetes of the young? Nobody cares, right? Is there a cool mouse mutant? Nope. There is one thing, and one thing that relates them all. For these ones, and these ones alone, the pharmaceutical industry made a chemical inhibitor and gave it away. A very good chemical inhibitor. So you set up the assay. If you recall, you can do so-called reporter assays. They put the DNA binding domain upstream of Gal4 driving something. They fused the ligand binding domain. They sprinkled the GSK library on. They found something that inhibited it. They optimized it to make a beautiful inhibitor of the protein of PPAR or FXR, and then they gave it away. And so then it became sold, so anyone could buy it. Anybody here could buy it for $80, right? In the catalog squirted on your cells. And look what a dominating impact this had on the biomedical research ecosystem. If there was a tool compound available that was anyone could have, it catalyzed researchers to work on genes and proteins that they wouldn't have worked on before because the bar just got lower. Right? And so we didn't decide to make inhibitors to make drugs. We decided to make inhibitors using the skills of drug discovery to discover what it is to be human. What do all these genes do? And to take advantage of all the biology around the world. The easiest thing to do is put it in the Sigma catalog so anyone can buy it anywhere and they can do an experiment. So what is one of these molecules that we make? They're actually pretty refined molecules. To date, they cost about $2 million to make. First, you have to make sure that the compound that is active at less than 100 nanomolar It has to work in cells at less than a micromolar. If you have to add more compound than this, it's inhibiting other things. So a selective compound will have features of 1 micromolar. We tested against many other proteins in the human genome. Compounds often hit a GPCR by accident, an ion channel by accident, so you can profile for those. That's quite common in the industry. Let's pretend you have one family member and it's related to another one. You want to make sure it's selective for the family member you want of proteins that are related and not the others. And you have to know that it got into the cells and hit the protein that you think it's hitting and stopped it from working. So these are what our organization defined and is now the community standard for a chemical probe. A molecule of this character you can use in your experiment and be fairly confident that it's hitting your protein, and the biological effect you're seeing is due to that protein that you think it's hitting. We often provide an inactive molecule at the same time, a molecule with a very slight difference that we know doesn't work as a negative control. So these are the chemical probes. They're complementary to genetics. So everyone says, wow, why don't you just do a knockout? Well, in a human cell, for example, if you do a knockout on day 1 and it has some perturbation, by the time you're measuring it, the cell has reached a new homeostasis state, right? Because you'll see a big effect and then transcriptions will start to move and it'll be in a different state, but your signal that you want to measure is this big. A tool compound, you add at t equals 0, the protein function just got stopped. And 5 minutes later, whatever, you're seeing the ramifications of that. It's also reversible. You can take off the compound and you better go back to what it was before. You can do concentration effects. So they are orthogonal and complementary, but there's a lot of advantages to chemical tools. But they have to be good, right? There are countless papers in the hundreds of thousands to millions where biologists have put molecules like these on cells, seen an effect, and erroneously linked the effect to a protein through these compounds that they thought were selective. There's a whole rogues gallery of these. The one on the curcumin, that thing you dump it on cells, no matter what you're measuring, you're going to see an effect. And look at that, there's 22,000 papers on it. Almost all of them are going to be artifact. Molecules have gone into clinical trials that have not hit the target. Sanofi put up what they thought was a PARP inhibitor into phase 3 clinical trials. That means it's being put into people. And at the end in phase 3, they realized, oh my gosh, it's a non-specific inhibitor and it hits everything. And of course, it didn't work. So it is really important to take the care to make these properly. And are they having the impact? So we launched in 2009 a huge program to start to make tool compounds to that part of the curve. One of the first ones we make was a molecule called JQ1. This was made from our lab at Oxford in collaboration with a lab at Harvard. And this is when we put the tool compound out. And look what happened. Just like those nuclear receptor things, all of a sudden on this protein, people started to work on it. And most of the papers came from the use of the tool. They weren't just random people. BRAF, that curve, the little blip I showed you before, it was sequenced and it was found to be mutated in melanoma. A tool compound was made and now everybody uses it. And you wonder, why BRAF? Why was it way down there on the curve? I asked Tony Poston, who was a professor in Toronto, Tony, it's a cool protein. Why wasn't anyone working on it? He goes, Al, because there was an ARAF. And often, if you look at any protein family, PARPs, 1, 2, 3, 4, 5, one's more than 2 on papers, one's more than 2. It's silly. They're named randomly, but people do their work. It's really bizarre. So if there's a family with A, B, C, I tell you, publications on A will be more than B, will be more than C. So as a result of this, we've made tool compounds. So each of these compounds here meets those probe criteria and are being used in cells all around the world. We donate the methods to use it to companies, and then they just sell them to multiple companies. So in one of these instances, we keep trying to make these chemical probes. And the Gates Foundation, why I'm here is the Gates Foundation brought us and a lab over at the CCBT together. They said, we need a chemical probe for some proteins that are involved in human fertility. and it's PGK2. This is narrowly expressed in the sperm. It's a metabolic enzyme. And the hypothesis was that an inhibitor of this enzyme could be used as a non-hormonal contraceptive, right? And so we ran chemical screens in Toronto, at Baylor, and at INSTEM, and the same sort of scaffold popped up. These are weak inhibitors. They can't be used. This was not 10 nanomolar at the time. The group here solved the crystal structure, and then we decided to continue the experiment. So when you embark on a drug or probe discovery program, you sort of classify on that side what exactly you want to do, and the team worked together to try and improve these molecules so that it would get into animals, would be able to not get peed out by the mice right away, achieve the right concentration of the compound in the right place. And so we've met all the criteria. This is now going to be, we have a process where if we make a candidate chemical probe, we go to an external committee and ask them to vet it, because we don't want to be the masters of our own decision. So we have an external SAB go this, like Nero, and sure enough. So it's been a great molecule. The lab in Bangalore did, I would say 70%. The chemistry was done by us, but everything else was done here. It's like a super cool collaboration, and it's going to be an interesting molecule. It's now we've given it to 10 labs across the world who work in this area of biology that we don't know how to do internally, and they're all testing it in parallel. We just gave it away. None of this, normally when you give away a compound, if you're a company, you get the compound, then you get the contract. The contract says, I own your firstborn, I own everything you do in your life forever and ever and ever, and then it goes on and on and on. Ours are, here you go, good luck. So that's it. So if you look over the last almost 15 years now, the probes we've made, a couple of hundred, all of them are published in cool journals. We've given away well more than 50,000. They've been cited a lot. And 100 clinical trials have started, not from the molecules we made, but based on ideas that the molecules generated. Somebody took one of our probes, squirted it on, let's say, a leukemic cell, and unbeknownst to all of us in advance, the cell died. And so the compound then says that protein, which is the target, is a good drug target for that. And then the companies go, thank you very much. I can make my own compound. I don't need, that's our core business, but I would have never known that that protein was involved in this. And we made that connection with the use of a chemical, but the chemical wasn't the aim of us to make that chemical for a drug. We got our big paper, we had fun, and then they could take it for the rest. But what happens, I often get the argument from industry, university, if you put chemistry in the public domain, Al, you queer the pitch. There'll be no drug discovery, no one will touch it, no one will invest. And so I'll give you some examples. These are some of the tool compounds. We made this one with Harvard, this one with Pfizer, this one ourselves, this one with Lilly. These are scientists inside the companies working with us to make these molecules to give away, and they were all given away. And based on these discoveries, other people said, wow, that's cool, we're going to keep going. And that's the molecules that are in clinical trials now, and I colored in for the non-chemists, which I'm not one. You can tell where they came from, right? So this was an open source molecule, right? And the company said, wow, that's neat. I'm going to just change it a little bit to make it more like a drug, not like a chemical probe. And then they did the clinical trials and stuff. So what we discovered was these are having a scientific impact. These lead to cool discoveries. They do not inhibit drug discovery. If anything, they increase the activity on the novel targets. So it's been a lot of fun. So on the one hand, we could say, high five, aren't we cool? On the other hand, we suck. I mean, we spent probably $400 million making these 200 probes, and it took us 10 years working with Pfizer and Novartis. And it's kind of, we want to do 20,000, we did 200. It's embarrassing. And so how are we going to do the 200 to 20,000? Well, the structural biologists in the audience, if I'd been talking here in 1989 about structures, right, we would have done 200 and everyone was a PhD student or an arsenal of people, and then all of a sudden AlphaFold, right? And the recipe on turning an intractable problem to a tractable one is given by that AlphaFold example where PDB, the Protein Data Bank, where all our structural data go, in 1971 it was created, the first database to share biological information. And then the governments funded careful annotation of those data, statistically parameterized file formats the same so that machine learners could download the data and it would be in a very sort of strict way. Then John Moulton in 1993 said if we're going to make algorithms to be able to predict structures, we need to compare them. So he started the CASP competition. Every 2 years, from 1993 to 2020, 27 years, writing grants to keep that competition going. He's a hero. And then in the middle of that was us, structural biologists, dumping data into PDB. $2 to $5 billion worth of data in there as a public good to let the algorithm. So AlphaFold was going to happen, right? We just didn't know when. So how are we going to do that? Well, let's do the same thing for small molecules. Let's create a database. Let's create a data. Let's run competitions. But this time, we're going to do it with the community and not let Google win. Okay. And so clearly the only way to get from where we are now to there is to use computational methods in AI. There's no way we can scale the experiments that we do in the lab. But what's lacking is quality data. And the quality data haven't been there because the pharmaceutical industry doesn't have it, for one, nor would they give it away. Universities think, oh, if I make chemicals, we should patent it. So by definition, they can't share it. So it needed a cool structure to put together to say, okay, we're going to do it. We're going to create data for the public good. We convinced industry to fund it. We convinced government to fund it. And now we're off to the races. And I'll tell you about that project. And I'll tell you how you can participate. So the plan is to say, let's just get a data set of 2,000 proteins on this axis versus billions of small molecules on this axis and measure their binding as a public good, right? And so we've made the proteins, so we have that. We decided to use assays that are generic. Imagine if you wanted to measure binding of small molecule protein, and for every protein you had to develop an assay, an enzyme assay or a cell assay, or you'd be all the time developing assays and getting nothing done. And so we said we need some assay that you can do the same thing for every single protein. So these two assays I'll tell you about are just binding assays. Does the compound bind? It doesn't tell you about function, it tells you about binding, okay? And then of course bring in the community under our terms of open source. Run benchmarking. So we started a collaboration with the leading benchmarking organizations to organize this, and then increase the scale without sacrificing quality. As a biochemist, there's got to be quality data or else you're going to get nothing. So the first thing is affinity selection mass spectrometry. And this is a simple method. You get compounds in a pool. So in your little Eppendorf tube, you have like 500 compounds, each with a different mass. You put your protein in, and it's histidine tagged in our experiment. And then you pull out the binders, throw away everything that doesn't bind, and then you have your protein bound to a compound. You add methanol, the protein denatures, off comes the compound. You squirt your methanol on an LC-MS column, and it says, oh, something bound to the protein with a molecular weight of 302.12462. You go back to your list of 500 compounds, that's it, right? There's a lot of false positives and things, but that's simplistically what it is. And so that's what we're going to do. We're going to screen these 20,000 against 20,000 compounds against these 200. In this experiment, though, it's something a little different. We selected our 20,000 compounds to be racemates. For those, remember your chemistry? If it has a chiral carbon, your compound, same chemical structure, but will actually be mirror images of each other, sometimes 50-50. And so proteins bind things in a chiral way. And so what we have is, in the library, we have the equimolar mixture of the two. We bind it to the protein, we wash, and then we measure which came off. And often it's just one of the two. A beautiful control, right? Because then you offered it both. The protein side, I don't like that one, I like this one. And so it's much less, you have to tie yourself in pretzels to see how that could be an artifact, right? And the toughest part of this experiment is checking that the binding that you saw is real. Okay? And so that's the flow. In practice, here's an example. This is an E3 ligase from human, one of these proteins. This had no chemical bound to it ever. We purified it. We mixed it with 20,000 compounds, and one bound, that. And we saw a peak. Okay, something with the molecular mass of whatever it is binds. Then we noticed that that compound, you run it on a different column, and you say, this is a column that separates the mirror images. You say, oh, look, in the library, we have the R and the S version. Interesting. But what eluted from the protein is only one of them. So that gave us, well, we should be pretty confident that that's not an artifact, not non-specific binding. But to rigorously show it, we screened this compound against, in the pools, against 40 other proteins. This is how much it bound to our protein versus the rest. So you have selectivity, and then you have specificity. Only one of the enantiomers bound. And then we purified each of the enantiomers, ran SPR. This is a binding technique that measures binding, and you can get a curve. And then saw the co-crystal structure and the chiral centers right there. And you can see that it could have gone like this, but it's bound to the one like this. And we've seen this now over and over and over again. So these data will generate 20,000 compounds. We'll have compounds that bind and compounds that don't bind, so that the modelers can play with those two data types. Okay, so every protein we produce will be done through that. The other method we're using is so-called DNA-encoded libraries. You've probably heard about these. They've been around for a long time. Industry is really the home of this thing because it costs so much to make these libraries well. We've managed to convince industry to give us some. And in this one, you take your proteins and you screen, again in a small tube, now 1 billion molecules, and I'll tell you how. And then the same experiment. You wash away the things that don't bind, so you have your negatives, and then you find out what does bind, your positives, so you have, again, a training set to train ML models. In this one, what you're trying to do is to use the ability to amplify DNA to display hundreds, millions, billions of molecules. So you get an oligo that's got a conserved bit here, and then you attach a compound, and you ligate on a piece of DNA. So this DNA tells you that that compound's there. Then you do a chemical reaction with a lot of different chemicals, and then add more. And then you do, again, take the purified, add more, and the kind of split pool thing that combinatorial chemists have done forever allows you to quickly go from one molecule to 7 million or so in these numbers. And so you have a little itty-bitty little test tube. It's in like 100 microliters, right? But you have 1 million molecules in there, and you'd never be able to detect them if you weren't able to amplify the DNA after. So what happens is that you take this mixture that's in your tube, you immobilize your protein, so ours is histidine tag, you bind it, you wash away the unbound, so these are all the chemicals that don't bind, and then you elute, you do deep sequencing. There's a little bit of mathematics in the middle to denoise it and decode it. You get your theoretical structures that you imagine there, and then you select compounds without the DNA. And why you do that is, this bound to your protein, this is real size. This is the DNA and the compound. So your protein's going to have opportunities to bind a lot more things than your compound, right? So what the industry customarily does is say, well, let's just synthesize this out without the DNA and test it. But synthesizing that costs $3,000, right? And there's so much noise that people would spend $3,000, take 2 months, doesn't bind. Spend $3,000, doesn't bind. And so it becomes uncost effective to do it that way. And so what these guys came up with, so we started work with these guys in 2018, and in 2020, Pat Riley's at Google, he was the head of Google Accelerated Sciences, and these guys were at a biotech company. They had the idea, in that Dell experiment, don't make anything. You have a bunch of compounds that don't bind, and a bunch of compounds that do bind. Could we use that to train a machine learning model on what the shape of a hypothetical compound that binds should be? A model of a binder. And then when you get that model of a binder, then you say, we can go purchase compounds that look like that. So an important thing to know is contemporaneously to all this, in Ukraine, after the fall of the Soviet Union, the chemistry department had no money. So they just started to make compounds and sell them. And they became very good at it. And fast forward to 2026, they can make 6 billion compounds that you can just buy for $80. Not $3,000 one, you can buy it for $80. So you can understand the experiment now. You do this binding assay to generate data. Positive training data, negative training data. You build an ML model of what a compound looked like, the pattern of a binder, and then you go, is anything in the purchasable, easy to get data set, catalog basically, and then you purchase it. And that stuff works. And it worked in the 3 test cases. They collaborated with us and we tested it on 20 proteins that had never been done before. And we got success for 8 of them using just machine learning to pick. We didn't do an experiment. Just purchased it, built a model, bought compounds, they bind. For the machine learners out there, it worked 8 times, but it didn't work 12 times. We had to predict 50 compounds to get them to work, and only 2 of those 50 worked. So you have this cool situation where it's a sweet spot of ML, where you know it can work, but you know there's a hell of a lot to get better, right? And once it gets better, what we'd love to do is to be able to just say, here's one structure, predict one compound that binds nanomolar. So we don't have to do any more experiments at all. And so again, the experiment is to take these building blocks, right? You've got various combinations of compounds. You bind, you do these sequence reads, you classify binders and not binders from these binding events. So customarily, there are 3 reactions. So chemical 1, 2, and 3 are attached together to make a shake. You notice that when you have this and this together, it binds. These don't bind. Then you classify them, yes, yes, no. Build your model, predict from this commercial library, purchase them, and then test them biochemically. And that cycle works, and we've done it quite a few times. Okay? And here's an example of some of them. These are the molecules. It happens to be a protein WDR91. We screen them at our lab at University of North Carolina. We build models. This was nominated from a machine learner graduate student. He wasn't a chemist. He just took the data, plugged it into his program, ordered this compound, and he was right. And other different folks did the same thing. So each of these are predicted compounds that show binding. So it can work, right? And what we did was we built a dead simple ML model to be able to set the benchmark. It's with this model that's been put up onto our website that anyone can download. We know this works in the simplest case. We know it didn't work in other cases. We don't know why. And so now we're into the process of trying to improve the model. Okay, so you can kind of see it's really simple. I mean, this is the simplest experiment we've ever done, right? 2,000 proteins, lots of things, generate data for the public good, and then sort of engage the machine learning community with the aspiration, any protein, find a compound in a day. We're far from there, but that's where we want to go. So these 2,000 proteins, where are we going to get them? So in an organization over the last 20 years, we've made 3,000 to 4,000 different human proteins. And we could make them all, but this is our... When I normally tell this story, I get to, but what happens, let's say, if there was a woman in Nebraska who's worked on a protein for 25 years. She's the world expert, and I'm going to change it to Bangalore now because I actually met the person I'm going to talk about. And she's worked on a protein for 25 years, and she's the world expert. Everybody wants to know about that protein comes to her, right? Why would we make that protein in our lab in Toronto when we could just pick up a phone? Hello, I know you love this protein. If you send it to us, we'll screen it with all our fancy schmancy methods, which would cost, FYI, a couple of $100,000 on the open market. And what we'll do is we'll give you back the hits if we find any. So you're no restriction, just play with them. But you've got to agree that the data go in the public domain. And we've said that to now 40 or 50 professors around the world, and every single one, I'm in, right? The data in the public domain, cool. Like most professors are lefter than Mao Zedong, so they, yeah, peace, right? and they get hits for their proteins that they couldn't get before. So this has been a really interesting thing. So the idea is that contribute your proteins to help us. We'll put them through industry standard methods. And any of the hits will be provided back to you without restriction. You can write grants. You can do anything you want with them, right? So give us your protein. We screen it. We put the data in air check. You get the hits. The hits are the exhaust of our experiment. The data are the asset that we're aiming for, right? This is a data project. If we happen to generate cool chemistry from your protein, go to town and have some fun. And so there's a website on the bottom right. You just click, and that's all you have to agree to. Peace and love. And somebody did agree to it, and this was Nat Moorman at UNC. He happened to be working on avian flu. He happened to be working on the PB2 protein. And so he sent us the protein, and we did the screen. We screened 8 proteins in parallel. So this is how much this compound binds to his protein versus the others. Selectivity. It was a racemic mixture, and this one had great preference for one enantiomer over the other. And so we sent him the compound top left, and then in a few cycles this is where we stopped, to be honest. We gave him this. It just so happens that he had a chemistry lab next door, and they did some chemical optimization, and he's got a sub 50 nanomolar and 300, all in about 8 weeks from beginning to end, right? And it's really interesting because this is all done in the open. I mean, he's free if they wanted to. They didn't file IP or, on the bottom left structure. That's your lab, or if it was you that sent it, or you could do it in the open. It's up to you. But the compound on the top, your top right, and the data are in the public domain. So this is, we're finding great appetite for this. And one of the reasons I'm here is that, unfortunately, currently, if you want to send me your protein, it's got to go in an airplane halfway across the planet, and the odds of it getting there in one place are between 0 and 0. And so somehow we have to get this capacity going in this country so that your country can contribute to this sort of global endeavor, and that means that the data collection has to occur here as well. In addition to the reasons that we all want to do it as a family together, but it's logistical, right? If we try and ship it to Toronto, odds on it's going to cost a lot of money, and the protein's going to thaw, and it won't work. The other important community to engage in is the machine learning community. It's fairly certain that we're not going to have machine learners in-house that can do it all themselves, nor would we want to. So how are we going to attract scientists to the data? Because these data are going to be the largest by orders of magnitude over what's out there already. And so we created a network. We thought, okay, how are we going to do this? Why don't we see if there's an appetite for people to join the project? We created a formal structure. It's called Mainframe. You go online, you click, you basically say, I'm going to open source my algorithm. I'm going to participate with you guys in your benchmarking challenges, and that's about it, right? And so what we're going to do is we're going to generate some of these data, keep them out of the public domain for 6 months, allow the open source community to play with it so Google doesn't see it, right? And then eventually it's obviously going to go out there for the world. But in the interim, we definitely want to give the open source people an advantage so that these algorithms can be built on. And if you don't want to open source your algorithm, you can wait. You can till the data are in the public domain, right? And so this is going to be fun. And so far, 230 scientists have clicked. We haven't really announced it yet because we haven't started to generate data. But here's a great example for students and faculty members here who are good in machine learning to come and join the project, get access to these data sets as they start to roll off the pipe, and build models. And for the top performers in our first round of competitions, we'll test your models prospectively. You make predictions, we'll purchase the compounds, and we'll test them in the lab. That's a $20,000 experiment, right? Because we want to give incentive for the best brains in the world. And why I'm so excited, well, one of the reasons I'm so excited about this, we did something like this. We gave, through a competition called CASH, we put a structure in the public domain. We said, predict molecules that bind, we'll pay. Now, we had to have a peer review competition to get in because it costs a lot of money to do that experiment. 25 people passed the bar, Harvard, Caltech, and the best person, one who had the best compound, came from there. A tiny little university in the southernmost state of Brazil. My Brazilian students in the lab hadn't heard of the university. I said, they're from this university. They said, well, no, that university doesn't exist. You mean this one? No, look. And they all went, I didn't even know that was a university. So can you imagine? We had Caltech, Harvard. If you were to say, I better find the best machine learner screener, no one would say, I know. Let's go to southern Brazil, to a university I've never heard of, to a woman I don't even know, and choose her. But when she was given the chance, she won. It was so cool. And so I'm imagining that what we're trying to do is that on steroids, right? Get these data sets out there. Get folks, wherever they happen to be, make the ability to compete as the difficulty in competing in these competitions as low as possible, and see what the best person wins, right? It's a communal effort. And then the algorithm, because they're open source, they can build off one another. You can compare computational pipelines to see which one's working. And that's kind of stuff's ongoing right now. So you can see, well, I'm going to go back one. So you can see my fantasy world, right? I have a rich fantasy world. And so what we're going to do is we're going to generate all these data. We have the funding for it. We're going to put these screening pipelines in place. We have it. We're going to get people from all around the world to send us their proteins. We're going to put them through. The compounds are going to go back to the scientists so they can do cool biology. And the machine learner is going to train their models on the data, make predictions, and hopefully in 3 to 5 years be able to predict without doing an experiment which compound binds to which protein. And that'll democratize that part of its really early drug discovery or chemical probe discovery, but it's the first step. Finding the first compound is the current hardest part. And we're going to do that as a community. And the last thing I'm going to talk about now, one more slide, and it's this one. Machine learners don't care where the proteins come from. We do. We're funded to do human proteins. Some of them can't spell the word protein. And so why not, if we get this technology that we built on the backs of a lot of pharmaceutical money, why not apply it to other systems? Complex microbial systems where you really want to know what a protein does, but you can't do genetics in it. Mixed microbial consortia, right, where they're obligate syntrophs and you can't culture any one of them. There's no genetic plasmids for them, so how are you going to do this system? Complex plants that are polyploid, that are really hard to do genetics. Find a compound that inhibits. If we can make this cheaper, more cost-effective, we can start to think about how these tools of chemical biology can be applied across biology. They've been in the domain of the pharma sector for so long, because these things, currently if you wanted to do one DNA-encoded library screen in industry, you'd have to pay $220,000 US. One protein, one screen, right? And so it's beyond my wife's an engineer. That's like her whole grant for 3 years on one screen, right? And so imagine if now we get it more cost effective, we have the funding, and I know people here work on a variety of different biological systems. Just think about, maybe if we get this operation going here in Bangalore, that maybe some of the plant proteins can go through, microbial proteins can go through, viral proteins can go through, plant viruses, animal viruses. We're not limited. It's just a protein, right? For the chemical library doesn't know where the protein came from. So I'd like you to think about, this for the students out there. Think about the applications that this might have, apart from, the human genome, which is really our focus. Okay, thanks for your time. I'm glad no one else mentioned ice hockey today, because it was a sad day for Canada yesterday. I won't mention cricket either, or South Africa, or anything like that. Thank you.
Thank you. It was wonderful. It's open to the questions to the audience. Anybody has?
Right now, for the proteins that you would be screening, I didn't understand. You have these 2,000 proteins. And so the other proteins that people would send you are covered under this, only if they're human proteins, or how does it work?
So for the questions were the 2,000 proteins, have they been selected, and are they only human, and does the proteins donated from the community count? Well, it's kind of an interesting answer, because 2,000 is a number we wrote for the grant. We just think we need a big number that sounds impressive, but there's no quantifiable way that 2,000 is the exact number. So I think that 2,000 is going to be inclusive of the proteins that people sent to us. So we're trying to get 500 proteins from the community. Among the 1,500 that we're going to do, we're going to look for diversity of pockets, right? So in proteins, there are binding sites, right? And if you look through the human genome, the people have gone through each protein looking for a pocket that has enough square angstroms to bind something, some hydrophobicity, so-called druggability. And so there's lots in the human genome. So there's whole groups sort of lining up how many druggable pockets there are. So we'll have to get diverse sampling of that space. There's funders that want their proteins. There's disease associations that want. So if you integrate all of that, it hopefully will come to around 2,000.
Okay, and your screens are binding screens, right? So all the stuff about cellular permeability and so on, those are not,
Definitely
separate.
Exactly. So as we go to 2035, once we get hits, then the next thing will be how do we optimize the hits, how we get. But we thought we needed a focus, so let's focus on the first step that's currently the hardest, which is finding binders. After that, we'll optimize the next one, probably starting in parallel. But you're exactly right, it's just a binder. It's not even functional necessarily.
Any question? Sandhya.
Any experts that have worked on a protein for a long time and are the world expert?
Does your chemical library include molecules that are already known to be drugs?
So the answer is no. Our compounds tend to be smaller. A drug on average is 450 molecular weight. Ours are about 350, because if you start too big, then there's no way to improve. So you have to start smaller than you will. And approved drugs have been optimized for purposes not binding necessarily to these proteins, because you've taken one of the hits and really refined it so it's selective. So you wouldn't expect that it would bind others. We have tubes in our ASMS screen of intracellular metabolites, because a lot of these proteins probably bind cool things through feedback inhibition that we haven't even thought about yet. Some tox compounds, but apart from that, no, not the known drugs at all.
Firstly, thank you. Fantastic talk, and learned a lot. What I was wondering is, when you're working with enzymes like the one which you showed about glycerol kinase. Now, glycerol kinase is going to be there even in the microbial system. It's going to be there in yeast and so on. So
it's two in human. They differ by one residue in the active site.
Yeah, and one of them happens to be some sperm
Exactly.
profile, etc. So how can you help the other communities, the microbial, the viral, and taking examples of proteins which are actually very ancient and likely to be present in human? Can we then get a jump start in trying to understand those enzymes or very, very...
Yeah, so the question was that some of these families are conserved in evolution, and they're actually conserved by the chemistry that they do. And it's a known strategy in the pharmaceutical industry. For example, kinase, you have certain privileged scaffolds, they call them, things that will likely hit many, many, many kinases. And those are so-called starting points to design in selectivity. And so when you're making a chemical probe, you start with a weak binder, micromolar. Then you sometimes with a co-crystal structure say, oh, there's a little hole there. Well, I'll put a methyl on to fill that hole. And that hole's in the PGK1, but not PGK2. So all of a sudden, it's beginning to bind one and not the other. And you keep arborizing the molecule to get it more selective. And so in your example, when you have proteins that are conserved across phylogeny, then you can absolutely do that. Start with your hit, which will likely hit them all, and then to design in selectivity that way. The PGK2, normally we wouldn't work on it because we kind of know what it does already. This was a specific one funded by the foundation. It's definitely not. Well, actually, PGK2 is on the long curve. PGK1 obviously is studied like you know what.
So the second question I had is, in your experience, what are the KDs that you're generally getting for the compounds that you identify from your library?
with DNA encoded libraries, it could go anything from 1 to 5 micromolar to single-digit nanomolar. The ASMS library, because it's smaller, we occasionally see submicromolar. We usually see between 1 and 10 micromolar. These cannot be used in a biological system. They're too weak, right? Remember those chemical probe criteria to ensure selectivity? 100 nanomolar or better, 1 micromolar in a cell. If you put a compound of this weak binding affinity on a cell, it's going to hit 100 proteins, and you'll be erroneously linking the compound to the protein you want, right? It's actually hitting, it'd be like doing siRNAs with a whole library of them and trying to pretend it's a hit.
So would it not have been advantageous in the long term to have had a good chemistry setup as well, affiliated to your...
We have. We publish about 120, 150 papers per year, and half of them are in chemistry journals. Our lab in North Carolina is all chemists, right? You're exactly right. That's the next stage, and certainly an associated lab beside, because, for example, when the people donate their proteins, we find them a hit, we say, do anything you want. I don't think there's one that hasn't gone, can you help? I don't know any chemists. What am I supposed to do with a hit, right?
Firstly, the permeability issue. How are you going to get into cells?
Yeah, so the question was, when you're a proper medicinal chemist, will be able to look at a compound, make derivations of that molecule with cell permeability in mind. There are rules that they know through decades of the pharmaceutical industry. Organic chemistry professors don't know that stuff. Medicinal chemists do know that stuff, right? And so that's the advantage of working with medicinal chemists. So the question is, yes, we need chemists. It would be a great idea to put it here.
I have one online question from you. What is the size of the protein binder and non-binders data already collected?
So on the AirCheck database, aircheck.ai, there are, I think, 20 data sets. But unfortunately, so we ran the DNA encoded library data, but the companies that we ran the DNA encoded library allow us to use only computational representations of the structures, not the structures themselves, the so-called SMILE strings. Our new project, we've convinced companies to give us open source DEL libraries where you'll be able to see all the data. And so that is going to start, the library is being delivered April 21st. So June 1st is when the DNA encoded library stuff really gets off the ground, and that'll just go up through the moon. And the ASMS starts April 1st. So the data sets that are there can be mined, can play with them. People are playing with them. There's a dream challenge going on right now on our data, but the real data collection will start with the open source libraries in April and June.
Correct. So, Smithcamp gave in 2008-9 those molecules, and then NIH opened up Francis Collins some molecules. But point is, your screening library, do you have all FDA approved molecules as well as all clinical trial failure molecules?
No, so we don't have, the question was, again, so there's a lot of compounds out there that have been optimized, been in people, and people hope against hope that these compounds can find other uses. That started in 2002, that idea. You know how many molecules have been approved for use after repurposing?
Zero.
Yeah, and lab-based assays when you do biochemistry. Now, drugs have been repurposed when a clever clinician noted that, hydroxychloroquine for lupus, when the U.S. Army was in Panama and they get them all for malaria, used to take it, the people with skin infections, hey, something's going on. It feels great. Right? And so they realized, there's an anti-autoimmune effect of this thing, and that drove the clinical. The Viagra is a similar story. They didn't think about it until they noticed something in clinical trials, right? So those types of repurposing happen all the time. The idea of taking an FDA-approved library, squirting it on cells in the lab, having a hypothesis, and taking it forward has systematically failed. There may be one example.
What I wanted to say, since you are investing on so many protein screening, see, eventually biology doesn't see one protein, one molecule. It goes into the human body, so many proteins, so many side effects. So actually your data, if it is screened on this, you might discover side effects and say why this protein, although it works for this job, but it creates this. And that is the new discovery.
Absolutely. That would be super
So
important.
that's the advantage of using FDA-approved drugs. And of course, the companies will get very worried. But what would be ideal if you can convince the companies to give you clinical trial failure molecules?
We can make them.
So
we don't
that
need their
one you
permission.
can- It's open. It is open. So
Yeah.
you can do that. And that, I think, is the future there.
Yeah, well,
This
we can do that here.
Second thing, I think your AI/ML model that we are talking about, I think it will be ideal that you can dream about, I like dreaming big. So you can have the AlphaFold, this tissue specific, the protein that is expressed in a particular tissue.
Yes.
And you know what are the gene expression of those tissue. So based on that, you can actually create an AlphaFold quantitative protein availability in that tissue based on the data, sequencing data. And then AI/ML guys actually can tell that this is going to work or this is not going to work, because you have a possibility of building that 3D structures there. So that's I will say that is the real situation. Getting molecules, machine learning is fine, this is very obvious. Open Source Pharma Foundation, they have done some work also, and they're part of your system. So this is where the opportunity is. How do you utilize this? Using AlphaFold data, create genomic sequencing, single cell data on tissue-specific expression, and use that quantitatively to see which molecule is likely to do in the machine.
And
I think that would be the real challenge, and you can do it. You have the resource to do it.
And where? Which region it's doing?
and which region as well.
Of the organism.
Yeah.
Give it to an
No. This is all human. He's talking about all human.
Okay, human swallows it. It's
Yeah.
come on.
No. He's talking
So-
about.
Ultimately reach the destination and work there.
Yeah. So it's an interesting, for the students out there, it's an interesting thing, right? You can think of so many things to do and your brain can explode, but finding a hit's hard all by itself. And so why don't we start with the first step, which is get it, but keep all this stuff in mind, right? Because there's so many ways this can go. But when you're running a project that like this has to be very organized, if you start getting distracted, I don't mean in that a pejorative way, but you think, okay, I got to focus or else we'll get nowhere. And so that's what we're going to try and do in the first instance. But the possibilities are endless.
Wonderful.
I was just wondering, what was the criteria for the 200 proteins that you chose?
The 2000?
No, the proteins are 200, and you said the compounds are 1,000. That is the eventual. As of now, it's 200.
Yeah, they're the ones we happen to have in our freezer, the ones that are structurally enabled, the ones that will generate data because we need to get the pipeline going. It's kind of boring, but under the hood.
Is that structures known, and they
Yeah.
they have cavities in them?
Absolutely, and because imagine if we screened 200 cricket balls, then nothing will bind, we'll get no data, the machine learners go do. Right, so we need to generate some data to generate the interest, and then we'll put more diverse proteins through.
I'm just wondering how many human proteins are there in the protein data bank as of now, which fulfill that criteria?
Oh, 4,000, I think. Yeah, or domains. Yeah. Yeah.
I think we should thank you for this wonderful talk. And
Thank you very much.
And excitement. And I realize, I didn't remember, but he remembered that we met 15 years back in Paris on open source drug discovery platform. I'm so glad to see that you are taking it forward.
Yeah,
Wish you all the best.
you're the inspiration.